In this video, I will discuss the structural functions of carbohydrates and sugar. Beyond its energy storage and energy-releasing functions, sugar also has important structural functions. In plants, the cellulose is the complex sugar that plants use to build their cell walls that give them rigidity. Plants, stalks, and the wood of tree trunks and branches are made of cellulose. In animals, chitin is the complex sugar that is found in the exoskeletons of invertebrates, including insects and crustaceans. Chitin is also found in small amounts in sharks, fish, and amphibians. Fungi use chitin to provide rigidity to their cell walls. Mollusks, such as clams and oysters, combine calcium carbonate with chitin to increase the hardness of their shells. Cellulose is a long-chain organic polymer. According to the reference book Comprehensive Biotechnology, quote, "Cellulose, the most abundant polymer on the Earth, accounts for approximately 35 to 45% of the biomass composition." Unquote. Living things are about 70% water by mass. If the water was dried out of living things, biomass could be measured in units of bound carbon. Measured in tons of dried carbon, the Earth's living biomass is about 550 billion tons. So, if 40% of this dry weight is cellulose, then the weight of cellulose on Earth is over 220 billion tons. Of Earth's biomass, about 100 billion tons is destroyed and regrown each year. The mass I quote for the living biomass does not include the mass of bacteria. The mass of bacteria may be equal to the mass of higher organisms. The mass of bacteria is uncertain. We cannot be certain how deep bacteria lives in the rock of the Earth's crust. The isomer for cellulose is C6H10O5. The isomer for cellulose is shown in the figure. Cellulose is formed from simpler sugars such as glucose and fructose that are chained together into long strands. The strands of cellulose are then cross-linked by hydrogen bonds to form a high tensile strength matrix that resists tearing. Cellulose is the material used in the cell walls of plants and algae. Plant cells have cell walls that are intended primarily to prevent the cells from ripping open when they are pressurized by the intake of water. By themselves, the cellulose cell walls don't have much compressive strength. Compressive strength is achieved by the water pressure, which causes plants to become turgid, or have turgidity. When plants become dehydrated, they lose their turgor. Their leaves wilt, and unreinforced stalks wilt or droop when the plant needs water. Plant cells make cellulose in their cell membranes in protein complexes known as rosette terminal complexes, RTC. The RTCs are hexametric-shaped proteins that are about 25 nanometers across. The RTCs float in the cell membrane, and each RTC will polymerize a long chain of cellulose that they use to build the cell wall by bonding simple sugars together using cellulose synthase enzymes. Many bacteria, including cyanobacteria, make their own cellulose cell walls using cellulose synthase enzymes. It is believed that the first eukaryote plant cells acquired the cellulose synthase they used to build cell walls from the chloroplast bacteria that they took in endosymbiotically to acquire their photosynthesis abilities. In woody plants and trees, the cellulose is cross-linked with lignin that provides the reinforcement that gives the cell wall compressive strength even when the plant cells are dehydrated. The lignin-reinforced cellulose makes grasses and the stalks of plants woody. The wood of trees and bark is cellulose reinforced with lignin. According to Bourgeon et al., quote, "Lignin is traditionally considered to be formed from the oxidative coupling of 3 monolignals, p-coumaryl, coniferyl, and synapyl alcohols." Unquote. Alcohols are defined as a hydrogen-saturated carbon atom with an attached hydroxyl OH group. Monolignol alcohols are based on the amino acid phenylalanine, so technically lignin starts out as a protein, not a carbohydrate. The monolignol alcohols are biosynthesized in the cytosol or cytoplasm of plant cells. The monolignol is linked to glucose to make it a glucoside that's soluble in water. The monolignol glucoside is able to pass through the cell membrane into the cellulose cell wall, the apoplast. In the cell wall, the glucose is separated from the monolignol. and then the monolignols are polymerized into lignin in the spaces between the cellulose fibers. Despite a century of research, the polymerization process of lignin is not fully understood. This is a limit of scientific knowledge. The lignin ratio in the cell walls increases as cells age, which make older wood more rigid. Young trees bend, while older trees stand up to the wind or loads until they crack or break. Even edible plants like asparagus stalks and carrot roots get woodier and harder as they age, and their lignin ratio in their cellulose increases. Lignin is insoluble in water, so soaking or boiling these woody foods does not make them softer. Lignin is also resistant to acids and strong bases. By itself, cellulose is an insoluble sugar. The lignin in the cellulose makes the cellulose even more water-repellent and water-resistant. The lignin allows leaves and bark to shed water. The water resistance of lignin allows the capillaries inside the plant stalks or tree trunks, the xylem and the phloem, to transport water or sap from the roots through the plant stems to places where water is needed for photosynthesis and growth. Both cellulose and lignin are indigestible to humans. Cellulose is digestible by some bacteria. These bacteria have genes to make the cellulase enzymes that can break down cellulose into its simple sugar components. Bacteria lack the enzymes to break down lignin, so lignin is indigestible to most bacteria. Only white rot fungus and its cousins are known to successfully break down lignin into simpler saccharides that other organisms might feed on. Termites prefer to infest wood that has already been partially broken down by fungus. Some termites do produce cellulase enzymes that can break down cellulose. However, termites cannot digest cellulose directly. Termites rely on protists and protozoa in their guts to digest the cellulose, including metamonad protozoa. These protists and protozoa produce their own cellulase enzymes. The protozoa break down the cellulose into simpler carbohydrates and fatty acids that the termites can use for food. Hoofed animals such as cattle, deer, goats, sheep, and other ruminants have complex digestive systems with up to 4 stomachs that contain bacteria and fungus that digest the grasses and leaves they eat, and break cellulose down into simpler carbohydrates and fatty acids. Horses are not ruminants. They don't chew cuds like cows and have only one stomach. However, a horse's small intestines can be over 60 feet long, and it contains protozoans and fungus that can digest tough grass cellulose. The process of digesting cellulose in leaves and grass is slow, and the nutrient content is meager, which is why these animals have such a complex digestive tract and must eat so much plant material. A 1,000-pound horse might eat 20 pounds of grass per day and wash it down with 10 gallons of water. Beyond its use as food for termites and horses, plant cellulose is used in many human industries. Many commercial products are made from cellulose or derived from wood or wood pulp. The wood products include lumber for building houses and furniture. Cellulose fiber can also be used as building insulation. Cellulose-derived paper products include copy paper, newspaper, and cardboard. Cotton is 90% cellulose. Cotton is used in textile products that include clothing and bedding. The pharmaceutical industry uses cellulose as filler in pills, and cellulose is used directly to supplement dietary fiber. The food industry uses cellulose as a thickener, emulsifier, or to reduce caking in processed food. The plastic industry can use cellulose as base material for making plastics including celluloid movie film, cellophane tape, and water-soluble binders and adhesives including wallpaper paste. When processing paper products from wood pulp, the amount of processing to remove lignin from the paper is dependent on its intended use. For copy paper and book paper that are intended to remain white for many years, extra processing is done to remove the lignin. For newspaper or cardboard, the lignin is not processed out of the wood pulp. Lignin is yellow-colored, which is why newspaper yellows with age. With cardboard and other packaging paper, yellowing is not a concern, and the lignin can be left in the wood pulp. The other important structural sugar is chitin. Chitin forms the basis for arthropod shells and exoskeletons. Chitin is used by some other animals and fungi. The exoskeletons of insects, crabs, lobsters, and shrimp are made from chitin. Snail shells and the beaks of octopus and squid are also made of chitin. Mollusks, such as clams and oysters, add calcium carbonate into their chitin shells to increase its hardness. Beetles and other armored insects toughen their shells and mouth parts with a composite of chitin and the protein sclerotin. Chitin is also found in small amounts in sharks, fish, and amphibians. Fungi use chitin to provide rigidity to their cell walls. Chitin is second to cellulose as the most common saccharide polymer on Earth. The monomer of chitin is acetylglucosamine, which has the formula C8H15NO6. Acetylglucosamine is more precisely called 2-acetylamino, 2-deoxy-D-glucose. Acetylglucosamine is glucose C6H12O6, modified with an amide NH3 group, and acetic acid CH3COOH. The acetylglucosamine monomers are polymerized, or chained together to form long chitin strands that are cross-linked to form a rigid matrix that is strong in both tension and compression. The figure shows the relationships between the chemical structure of cellulose and chitin. Chitin is effectively cellulose with amide and acetic acids attached at each glucose. Animals that eat other animals, or fungi that contain it, must be capable of digesting the chitin, or else passing it out of their digestive tracts. Animals digest chitin with chitinase enzymes. It is found how effectively the animal species can digest chitin depends on how often the species eats other species that contain chitin. For example, cattle often ingest insects that live in the grass they eat. Bovines such as cattle have chitinase enzymes in their stomachs that can digest this insect chitin and break it down into simpler sugars that add to their nutrition. In their daily routines, dogs will often eat insects and have no problem digesting the chitin in the exoskeletons of the insects. It is believed that both bovines and dogs have bacteria in their guts that produce the chitinase enzyme that can break down the chitin they ingest. When researchers Eri Tabata and colleagues studied several omnivorous mammals that might eat insects, they found chitinase enzyme ratios of mouse, chicken, pig, bovine, dog equal to 1.7, 0.9, 0.25, and 0.1, meaning that mice, chickens, and pigs would probably be able to digest more chitin or eat more insects than cows or dogs. This ability to eat insects reflects on their hypothesis of how often insects appear in their diets. At the opposite end of digestion, plants can detect that they are being chewed on by insects with chitin detectors. Upon detecting chitin, a plant will unleash its systemic defense system, which might include releasing toxins that make its leaves taste bad, or even poisonous to insects that are attempting to eat it. In people with seafood allergies, it is often that they are suffering an immune response to the chitin in the shellfish. When we look at the fossil record to see where chitin might have appeared, we find that the oldest direct evidence of chitin occurs in a 25-million-year-old scorpion that was trapped in amber. However, trilobites are arthropods that lived over 520 million years ago during the Cambrian period, and most likely made their exoskeletons from chitin, as all modern arthropods do. Trilobite fossils that old have had all their chitin replaced by stone and non-organic minerals, so there is no direct way to test them for chitin. In China and Australia, there are fossil beds of Cambrian bivalves that date back to 540 million years ago, and are even older than trilobites. It is very likely that these bivalve mollusks made their shells from chitin. Commercial uses of chitin include agriculture, food processing, and paper processing. In agriculture, chitin from marine shells and insects can be used to enhance fertilizer and as soil conditioner. In food processing, chitin can be added as a thickener or food stabilizer. In paper processing, chitin can be added to the paper pulp to make the paper product stiffer when it dries.
And that's my take on the structural sugars, cellulose and chitin. Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.